In Chapter 24, we're going to discuss the transcription of RNA and the post-translational processing of RNA. So the outline for the chapter is that we're going to talk about RNA. We're going to review or discuss the discovery, where our main focus will be on the transcription in prokaryotes. Then we'll discuss the modification in prokaryotes, and very briefly we'll cover the transcription in eukaryotic cells. So those two will be different, and so we're basically going to work through these. So this is part 1 of chapter 24. So transcription. Just as a reminder, DNA is the information of life. But what we don't see is a direct utilization of DNA to create protein. We first need to pass through the RNA phase. So in this case, we've discussed the replication of DNA. Now we're going to discuss the transcription into RNA. Eventually, and after this chapter, we will discuss the translation. I strongly encourage you to watch this video. There's a link in our module for this. And so if you remember, it's going to give a general overview of how transcription occurs. So what is the genetic code? So we've been discussing genetic codes for several weeks now. A gene is a DNA sequence that will encode for a protein or other cellular machinery. And so we find these in the genome. And so I think one of the issues is that students often believe that genes must encode for proteins. Some genes encode for additional functions. And so other genes will create other types of ribosomal entities, or they can encode for different other small molecules. So in chapter 25, which is the one after this, we will discuss how DNA sequences and the RNA sequences relate to the proteins that are utilized. In chapter 21, we learned that the total number of genes varies from organism to organism, but the structure of genes is going to be the same. So RNA has 4 unique bases: adenine, guanine, cytosine, and uracil. So 3 of these are consistent with the bases we observe in DNA. However, uracil is different. So uracil is not found in DNA. It is only found in RNA. And so the other thing of interest is that RNA is single-stranded. It still looks very similar. We have the 5' phosphate as well as the 3' hydroxyl. And so these look identical. We are just in a single-stranded entity in this case. And so they look quite the same. So the bigger difference when we consider RNA is the addition of what we're going to call this 2' hydroxyl. So this 2' hydroxyl is different in DNA and RNA. So in addition to a singular base change, we have an addition of the 2' hydroxyl. So RNA is significantly less stable than DNA. So the question becomes, how much different is it? So this 2' hydroxyl decreases the stability from many, many hours to lifetimes, if you want to consider the Jurassic Park phenomenon, to RNA, which an entire sample can be degraded and unusable in less than 2.5 hours. In 2 to 3 hours, it's all going to degrade. So RNA hydrolyzes in acidic and basic solutions. So if we were to look at this mechanism here, one of the things that we'll see, I'm going to remove this note here. So we can really look at this mechanism. So what we see is that we have our acid group or a base group come in, and we're going to abstract a proton. This proton is going to leave an O-, which attacks a phosphate group. So once we have this, we move into a cyclic intermediate. So once we arrive at our cyclic intermediate, it can collapse, leaving the 2' or the 3' nucleoside. So one of the things that we really want to look at here is the difference in these. And so there's a couple of entities or issues in this case. When we collapse into either of these are permanent, and there's really no way for the RNA to go back together. The formation of our cyclic intermediate and the collapse of that prevents the RNA from being useful. So we have completely degraded the RNA into separation. The other thing of note is that we now, in this case, would have a phosphate RNA phosphate plus another one that just has an OH, RNA OH. So what we see over here is a change in the stability of RNA. This mechanism continues. It can degrade randomly throughout the entire sample, and it will degrade the entire sample in several hours. So in the 1960s, Jacob and Menard predicted the existence of RNA based on a series of starvation experiments in E. coli. So E. coli has nutrients. These nutrients tell the cell how it should operate. In these experiments, what they did is they specifically targeted the cells to not use the sugar molecules that they are the preferred nutrition. So what we realize is through these experiments is that there is a tight control of the genes or the functional proteins at any given time. We're going to talk more about the operon model in chapter 26, but in this paper in the 1960s, this is really the first time that people predict that between DNA and proteins, there is a transient molecule. They don't quite know what it does, but they were able to say there has to be a control. So RNA, has three different types. We have tRNA, we have rRNA, and mRNA. So the difference between these, the tRNA is utilized in protein production. It carries the amino acids to the translation machinery. rRNA makes up much of the ribosome. So the total RNA is mostly based on ribosomal RNA. And messenger RNA, which is a small unstable molecule. And their lifetimes are short and can be detected by radiolabeled experiments. But first, they were able to determine different sizes. So RNA subunits are often called the 16S, the 44S. These are Svedberg units. And so these are a type of size gradients that we see in a size centrifugation. And so they were labeled based on their order and size. And so these tell us about the size of the ribosomal RNA fractions and how they are set up. And so we'll see this throughout this chapter. So in E. coli, we see all three steady states of these. We see all three of these types of RNA. So the rRNA is 83% of the steady state, but is only synthesized at less than 60%. tRNA is relatively consistent, around 10%, both at the steady state level and for the synthetic type capacity. But mRNA is only 3% of the available RNA content at any given time. However, it accounts for 32% of the synthesis. So the majority of the RNA in the cell is the rRNA, but the majority of the mRNA synthetic capacity is roughly a third of the cell's energy. So it tells us that mRNA is not very stable, but requires a significant amount of energy in order to create this. So mRNA. We'll talk a lot about mRNA today. And so messenger RNA is a direct template. It's suggested by Francois Jacob in 1961. It's very unstable. So in bacteria, the mRNA is degraded within 2.5 to 3 minutes, which tells us that the time between mRNA production and protein production is going to be quite short. So RNA synthesis is synthesized by RNA polymerase. It's a holoenzyme complex that utilizes a DNA template to create RNA. It's an oligomeric protein. It only has 5 subunits, which is different than DNA polymerase, which had a 10 subunit complex. The general mechanism, if you want to think about the arrow pushing mechanism for the way in which bases are added, is going to be similar to that, if not identical, of the one in DNA and RNA. And so if we look at our protein, we can see we have DNA over here in red, our polymerase, which is polymerase 2 of the 5 we met in chapter 22, and we have our RNA that's produced over here in green. And so we can see that in this case, the RNA is going to be produced from the DNA as a template. So the 5 subunits that we want to learn about today there are 5. So we have the alpha, beta, beta prime, sigma, and omega. And so the sigma subunit is going to be utilized in recognition. The alpha subunit is where we have active polymerization. And so there are multiple entities in here, and some of these, unlike DNA polymerization, will come and go from the replication process. So RNA polymerase is composed of 2 alpha subunits, a beta and a beta prime subunit. So this is the main entity. Both omega and sigma will kind of come and go. So these are transcriptionally active but not regulated. And so the regulation is going to come from the ability of determining the location in which genes are transcribed. So the sigma subunit is the promoter recognition. The sigma subunit is going to tell the RNA polymerase where to be active. So in addition to DNA polymerase, we are going to again utilize a magnesium that's going to be very important for polymerization. We can see in this case, in this figure here, we have the upstream DNA, and we have the downstream DNA. So we're going to be reading, that is unclear. Let's change how that looks. So we're going to read, we are going to polymerize, here's the 5' end, here's our 3' end. And so our gene will be read in this direction. So we can see that in this case, we have transcribed RNA being produced. So RNA polymerization transcribes at roughly 30 to 85 nucleotides per second, which is slower than we saw for DNA polymerization. The error rate is roughly 1 in a million bases, which tells us that there is a proofreading mechanism. Unlike DNA polymerization, the exonuclease site is contained within the active site. So what this tells us is that we don't necessarily unwind, but in the event that something is incorrect, the offending bases are immediately removed. So there are 2 magnesium ions that are required in the active site for polymerization. These are going to function quite similarly to the ones in DNA polymerization, where they are orienting the incoming bases. So when we think about RNA synthesis, we want to consider thinking about the sense and the antisense strand. So if we have a 5' to 3' set of DNA, the top strand is considered the sense strand, and the bottom strand is the antisense. Now these are quite relative in this case, but the reason the top strand, or the 5' to 3' strand, is considered to be sense is because when we read it from left to right, it reads in the order of which we will observe it in the protein. The antisense strand, you will have to translate or convert into a sense strand in that case. So as we read them in 5' to 3', this is read in the order in which we see them here on this top strand. On the bottom strand, it is the inversion of that. So when we think about this, we have the coding strand and the template strand. So we read, or we make a direct copy using the Watson and Crick base pairs of the antisense strand, which will allow our RNA to be in sense. So in that case, what I'm saying is that the RNA will read in the order and it will match the sense strand. So before the start of the gene, so if we have a start here, prior to that, we're going to start here before the end of the gene, if this is our gene, and it ends after. So there will be additional mRNA fragments that correspond to DNA outside of the gene of interest. So how does this all get started? So RNA polymerization is a selective entity, which is different than DNA replication. In DNA replication, what we saw was that the entire sequence of DNA would be replicated. And in this case, we have to assemble in the correct location. The sigma subunit's job is a promoter. And so it associates, it brings all of the other components together. And so they must all assemble on the DNA in a place where we can see the production of mRNA that corresponds to a gene. And so we're going to look at these two figures over here on the right several different times. And what we'll see is that we see there be 4 regions. And we're going to have what we call the open complex. So transcription occurs prior to the start of the gene. And so in this case, our green diamond here, this is going to be the start of the gene. Or in this case, the ATG site. So the promoter, or the purple star, is the DNA sequence that indicates a gene is to be encoded soon. It is what's considered upstream of the gene of interest. So the sigma factor is an assembly molecule. It recognizes a promoter ring region and starts the assembly of the RNA polymerase. Once the complex forms, it's going to dissociate quite slowly, on the order of 2 to 3 hours. So it tells you that the assembly is going to be very important. And so these are some of the identified regions. And so there are several different subunits that we can look at. And so these are, in this case, the different sigma subunits are active in different times in an E. coli's life. So in stationary phase, you see sigma 38. Stationary phase is after active growth. When bacteria undergo heat shock, the sigma 32 is going to turn on, allowing the bacteria to know that something different is going to occur. So there are 2 main promoters, one of which you've likely heard about, the Tata box, which is at the -10 region. Before that, you would encounter the -35 region. So these 2 regions are highly conserved and upstream of the gene. So if you look at this figure, we see the ATG start codon, then we have a -10 and a -35 region. So these are highly conserved, and they're often conserved across bacterial species and upstream of most genes. So when we talked about genes or the synthesis of genes in genomes, most genes are identified by the ATG start codon. Currently, there's been a movement to identify genes based on the -10 and -35 regions, which tells you whether or not a true gene will be transcribed. So our graphic has updated to include 2 additional portions. We now have the -35 region and the -10, the Tata box. The -35 region is also AT rich, but it doesn't have a name. It is not quite as repetitive as the Tata box, that is the -10 region. In video 2, we will start with the initiation.